past webinars have really focused on the benefits of dam removals. We've talked about, the importance of removing these obsolete dams for wildlife, for water quality, to restore the ecosystem, the different recreational benefits that come from these dam removal projects. But today, we're going to focus on another realm of dam removals that we haven't really touched on in these information sessions. And so today we're going to talk about the water quality monitoring, or the data collection that's involved in these dam removal projects. So we have a couple members from the partnership on the call today that are going to highlight an array of different topics. For those who don't know me, I'm Michelle de Blasio. I'm the Watershed Restoration Coordinator for the Nature Conservancy, and I help to lead a number of water quality monitoring programs in the Pollenskill watershed, centered around our restoration projects, but also our dam removal projects. And so I'm going to kick things off today and just give a very high-level overview of the types of things we monitor and look for, to measure these pre and post impacts of the dam removal projects. And then I'll turn it over to some of our colleagues also in the partnership that work to collect we'll dive a little bit deeper into some of these components that we'll touch on today. So when we think about the impacts to water quality, dam removals, right? So there's going to be, when we have these dam removal projects, we're going to have a lot of changes in the system. And so I like to say not all dams are created equal, but they're all going to impact the river and the surrounding ecosystem to some degree. And so depending on, the size and the scope of the project, those changes, the things we measure, those will vary. But you see here in this top photo, this is the backwater impounded area behind a dam. And this is what it looks like, right, when we take the dam out. We're restoring it back to this natural river, this natural riverine habitat. And so some of those physical changes we can measure, we can visually see them, but we can also take measurements to really assess, the degree of impact, the degree of change. Something that I like to note is those physical changes, both in the stream, in the floodplain, on the riparian area, but also to the sediment transport. Now you have a free-flowing natural regime restored back to the system. In the water column, there's also things that are going to be changing specific to the water quality. So you have those physical impacts, those physical changes that you see, but then the things that we can collect and measure are those chemical or the chemistry of the water. And so thinking back to, just keeping this visual of this lake-like environment that's now changing to a river-like environment, you're going to have a number of different things change to the water quality or to those very specific parameters. So for one, you have temperature. So when you have that very stagnant water behind a dam, like a lake, it's going to be very warm water temperatures you're going to see in that environment. When you remove a dam, you're going to restore the free-flowing natural flow to that system. Thereby, you'll be cooling down the water. You'll have trees growing on the banks, and that will help to also decrease or improve those temperatures. Dissolved oxygen is another parameter or something we can look at to show the success. So again, when you remove the dam and you have this free flowing, you're going to be adding more oxygen back to that specific stream reach, that specific water column. Turbidity as well, the sediment transport. Turbidity, for those who maybe aren't as familiar, it's just the measure of the cloudiness in the water. And so that is something that we can also take a measurement of, as you would expect. An immediate removal, you're going to have a lot of turbidity, but over time, when the river really returns back to that more natural state, that natural flow regime where it's, dropping sediment out in a more natural flow, you start to see those levels kind of even out. And now all these different chemical parameters and the different physical parameters that we look at, those all have an impact to the biology, the critters, the aquatic organisms that also live in the stream. So we also can measure or look at the resident or migratory fish or other aquatic organisms within the stream column to better understand how the system's changing pre and post removal. So there's lots of different things. My take home here, again, this is a very quick overview, but the take home is there's lots of different things to measure. We can really tell the story, the success story of these dam removal projects and the benefits to the ecosystem, to the critters living in the stream, to the surrounding area by collecting scientific data. Now, if you have a dam removal project that you are looking to monitor or collect information, where do you start? There's so much to measure. There's so much to look at. There's only so much time. There's only, so much time and money, right? So what I wanted to just kind of quickly orient folks to today is a number of different things we've created online to get folks oriented to, if you want to start collecting scientific data or start monitoring your project in some sort of capacity, a couple things you need to consider. So first and foremost is, what would be your monitoring goal? What are you looking to do? Do you want to show the impacts, the direct impacts to water quality? Do you want to show how recreational users are coming back to the area now that you've removed the dam, right? So there's a number of different monitoring goals that you could have. Some of those monitoring goals may be required. You might have a regulatory component or a permit condition that requires you to, track or measure the sediment, or you might have a surface water quality condition that's not meeting a regulatory threshold for temperature for fish, right? So there's a number of different things that you can set your goal to be. There's things that are going to be required from our regulators, but then there's also things like we call that would just kind of be nice, right? So you want to show to other funders the success of your project through the data, through the science. And so those monitoring goals will kind of shift depending on your specific project. But I think most importantly, it's how much time you have, how much funding is available. Some of these data sets can get pretty complex, and they can become pretty daunting to collect and manage. Other things will be a little bit easier. So again, these are kind of this is just a very high level of things to think about and consider if you do want to start monitoring your dam removal project. Now, in the Statewide Dam Removal Partnership, we have a monitoring work group, and within that work group, we've been working to kind of create cheat sheets for folks that want to consider monitoring their dam removal projects, but don't really know where to start, don't really know what kind of resources they need. And so what I wanted to do today was orient you to those resources, so you have kind of a one-stop shop for that very high level overview of all these different parameters you can collect, and then a little bit of a deeper dive or which partner you can connect with to find out more information. So we've created what we're calling this universal list of monitoring parameters or metrics. And then within those worksheets, we have defined the monitoring goals and acceptable methods for collecting that information. So this is just a very quick breakdown of all those different metrics. And you can see here what we've done is we've created the buckets of, what are going to be the parameters that will be required by permit? What will be the ones that, might speak to a surface water quality condition? And then most of the parameters, most of the stuff that we look at is project related or would be nice or research related. So you're not on the hook to do it. It's not required by the permit, but maybe a certain funder wants to see those benefits or those improvements to the water quality. And so what I want to do now is bring folks to the njdams.org page and just show you where you can find all these resources as they relate to these very specific parameters. So when you log onto njdams.org, for those who are not familiar, there's this tab up top. And when you go to how to remove a dam, there's an entire tab just for the monitoring page. And within here, you'll see, so a couple of the things, we have a couple guides that were not created by the partnership that are just available online resources that you can find there. We also have more information about, what does that mean required by permit? What does it mean, the surface water quality standards? So you can find those embedded within this web page as well. And then here we have each of those metrics, each of those parameters has its very own cheat sheet, if you will. So you can dive a little bit deeper into these, and just to give you a sense of what you'll be seeing. So chemistry, this speaks to the dissolved oxygen or the temperature, things I touched on earlier. And so this will just give you a very quick overview, what it is, why we monitor it, who would monitor it, is it required by a permit condition, or is it just something that would be nice to know to share with a funder or a project partner. And if you want a little bit more information, each one dives a little bit deeper and you get more narrative, more resources, more links. There's a ton of information. Water quality monitoring is very complex, but it's very useful. And if we need to have the science that really shows the benefits to water quality and wildlife habitats to make these projects successful and sustainable. And so I'm going to go back to my PowerPoint if I can. And with that, I wanted to just say, again, go to njdams.org for more information. I did a very quick run through of a very complex, very deep, there's tons of different things that we can talk about. If there was anything that you wanted to know more about that I did touch on today, pop it in the chat. We'd be happy to have a deeper dive into any of these specific parameters in future webinars. We're always looking for new ideas. But because we have a couple other folks on the call today that are going to dive a little bit deeper into some of these topics, I want to turn it over to Jake. He's going to talk a little bit about the sediment management and monitoring that is required. And then we'll turn it over to a couple more folks on the call. And I think we're going to save all questions for the very end, if that's okay. I'm going to go ahead and stop and turn it over to you, Jake.
Okay, thanks, Michelle. Can everyone hear me? All right. We had some technical issues earlier, so I was just verifying. All right, so yeah, I guess I'll just introduce myself and then I'll share my screen. So I am Jake Helminiak. I'm a hydraulic engineer with the Corps of Engineers, Philadelphia District. I've been with the Corps about 6 years, but I've spent about 13 or so in the private sector working mostly on dam removals and fish passage, river engineering, that kind of thing. So that's a little bit about my background. I'm going to go ahead and share my screen, and I feel like I have too many slides, so we'll get started. And all right, Michelle, can you see the presentation? Good. All right, perfect. All right, so like we said, I'm going to talk a little bit about sediment monitoring for dam removal. Again, so we have about 15, 20 minutes or so. There's going to be a lot of information, but this is kind of just a general overview of some of the approaches we use and that kind of thing. But let me get into this. So here's kind of a map of the presentation we're going to go through here. I'm going to give a quick primer about sediment management within New Jersey dam removals. Again, very high level, just to kind of set a framework for the stuff we're going to be talking about, and then move into the different approaches that we can use to monitor some of this sediment. I'll leave you with some references and guidance documents, just a slide on that. And then hopefully we'll have some time for questions, or like Michelle said, we might just save those to the end. So let's jump right in. Quick primer on sediment management. So why do we care about sediment? Why it's typically one of the main drivers for dam removal projects in terms of design and implementation cost. So I wanted to start off with common management concerns. So again, kind of the theme is why are we caring about this? So in 2016, a group of researchers kind of synthesized these common management concerns from dam removal projects all across the country. And again, I'll provide a reference later, but their research kind of came up with a broad suite of 7 or so common management concerns, which is, I'm not sure if you can read that on the right, they're a little small, but essentially kind of boils down to, I summarized it in 2 bullets, but kind of essentially reduced to impacts to local infrastructure, so such as incision of the upstream reach at bridges or pipe crossings, that kind of thing, or downstream aggradation at, say, water intakes or other infrastructure downstream, and B, impacts to water quality and habitat. So things like downstream turbidity, Michelle mentioned before, and aggradation downstream, potentially affecting water levels and that kind of thing. So these are questions that come up on nearly every dam removal project, right? So they need to be answered either qualitatively or quantitatively if it's a little more intense. So Moving to the next bullet, regulatory framework. I just wanted to, again, a high overview of how things work generally in New Jersey. So for dams that are larger than 5 feet in height, New Jersey DEP, Division of Dam Safety, will typically be the lead permitting agency. We might even have some of their folks on the call today, so they can feel free to correct me if I misspeak here. So there's lots of requirements within their regulations, but with respect to sediment, there's essentially three, boils down to essentially three. One, we cannot negatively impact flood levels. That's kind of a general one, not just with sediment, but two, we must consider the stability of the impoundment. And three, we must consider habitat impacts. So the habitat impacts are not specifically within the dam safety regulations, but they often arise through other agency reviews as part of the permitting process. And again, so must consider stability of the impoundment. It does not necessarily mean that we have to rock the entire upstream extent or remove all impounded sediment. These projects are, after all, in the name of river restoration. So we would like to leave the system on a better kind of ecological trajectory than we found it, right? So, and in fact, the remainder of the presentation assumes that we are releasing, some sediment and are allowing the upstream impoundment to more naturally adjust, also known as kind of passive sediment management approaches. In reality, there's almost always a balance of, hard stabilization or naturalized stabilization measures, and also with natural sediment movement, which is going to vary from project to project, based on specific constraints and those management concerns. All right, next bullet, the pre-removal analysis. So it just generally, I was meaning to kind of cover all of the data collection and engineering analysis that goes into dam removal planning and implementation. So these include, things like sediment characterization, identifying potential impacts, engineering analysis and design to deal with those impacts, and kind of ultimately coming up with a sediment management strategy or plan consistent with permitting requirements, kind of taking all that stuff into consideration. And also, it can be iterative. It may be iterative and maybe should be iterative, kind of using the engineering techniques and the permitting and kind of going back and forth to kind of land on a path forward strategy. So then we kind of, moving to the next bullet, we kind of say, okay, let's combine that in how we're going to monitor these things. So some of the best, so monitoring plan, I wanted to say, so it's often combined with like an adaptive management plan as well. So that kind of document would compile all the parameters that we'll be monitoring, how specifically we'll be doing that. Most effective monitoring plans that I've seen have been, very specific about goals, managing expectations from all the agencies, all the parties involved, including like when the monitoring, the parameters and the things that we're monitoring, when those, if they cross thresholds, when they will lead to on the ground, decision making and that kind of thing. I did want, kind of echo what Michelle said, that sometimes funding can be a little difficult for monitoring, but when we do have it, it makes it all the more important to collect when we can, particularly those data, that we can only collect during the actual dam removal itself. So once that's through, we can't go back in time and collect those data. So that's a consideration. All right. So I know that was a very brief overview. That was a lot of information, but we're going to jump into some of the sediment monitoring approaches that we can use to, again, assuming that we have worked all this out and we are allowed to, allowed to release some sediment downstream, and we want to understand how this is naturally, how the system is reacting. So we'll move through those. So just this list, we're going to move through. You know, baseline and documentation is not really a monitoring approach, but I wanted to kind of touch on that a little bit. You know, some of the baseline data is going to be, it's going to be done while you're doing the engineering and design and permitting, that phase. And it's just, you know, it's critical to allow the relative comparison to conditions during and after removal, right? So we have to understand what it is currently to understand how it's changing during removal and ultimately on that better trajectory, like we said. So I would make a case to really document everything. So not just all the successes of our monitoring plan, but, you know, the good, the bad, and the ugly. You know, it's all useful data. You know, oftentimes, in my experience, the unintended consequences or flops are far better teaching opportunities than successes. I don't want to say anything else about that. Oh yeah, so we really should start collecting as soon as we can. As soon as we have a project that we know is going to move forward, we should start collecting this baseline data. There's no time like the present to start collecting data. So next, so this moves into, again, starting with the actual physical approaches that we're using. So traditionally ground survey, so monumented cross sections. Again, so cross sections will be perpendicular to the stream. I know we have a range of folks on the call, so I'll try not to stay or be too technical. So cross sections would allow us to monitor things like aggradation or degradation within that cross section or the reach that we're talking about, widening, narrowing, you know, those changes in cross sectional area, those would be picked up there. Longitudinal profile, again, would be longitudinally down the stream. They would be useful for monitoring things like head cut migration. Again, in a natural sediment release, we would see, you know, base level drop and a head cut kind of migrating upstream through that impounded sediment. So monitoring that, changes in slope, other hydraulic characteristics. So all of these things, these physical things that we're surveying in the field, they would all translate into changes in sediment transport capacity and potential, kind of informing how things are currently changing and how we think that change will continue. You know, will things speed up? Will they slow down? Will they kind of stagnate between events? So you know, that kind of thing. So aerial survey. You know, we can also, so besides on the ground survey, you know, where we're out there with traditional measures, we can also use repeat aerial surveys to extract kind of the same thing, both cross section and longitudinal profile, topographic changes, that kind of thing. You know, large scale kind of LiDAR or aerial efforts are unlikely to fall within our smaller dam removal project timelines, but, you know, project specific data collection in these forms, you know, aerial surveys, use of drones, you know, that kind of thing, you know, it's becoming a lot more accessible and more affordable to do. So we'll be seeing more of that kind of project specific, you know, blanket of data more and more. One consideration, obviously, with this is that, you know, the underwater bathymetry is difficult to obtain with these methods. They are getting better. They can, you know, reach maybe a couple feet into the water in some circumstances, but just a consideration. And in those cases, if you have deep channel that you need information on, you know, these are not kind of independent. We can always combine different methods, you know, survey in the channel with aerial survey outside the channel. You know, one bonus of these aerial methods is that you also get an aerial photo of your site. So it's kind of a 2-for-1 there. All right, moving forward, stream bed composition. So, you know, understanding how the bed is changing, so grain size distribution, you know, we can get at that with a couple different methods. We show a couple things here. So pebble counts, where we're physically counting, you know, in a statistical way, pebbles and cobbles within the stream channel itself. We can also use you know, collect samples with a shovel. You know, I'm showing a gravel bar there, and then analyze those in the lab. So we can come at this, you know, multiple ways, but you know, we're trying to answer, you know, essentially understanding how bed gradation is changing through time, and that would answer questions like, is the downstream bed fining as the sediment pulse moves through the system, which is, you know, fairly common? Is the upstream reach coarsening as those fines are washed out of the impoundment due to increased velocities and sediment capacity, that kind of thing? So, and really we're documenting changes to these hydraulic characteristics, like roughness within the impoundment or downstream, which in conjunction with kind of some sediment transport information is kind of critical to understanding how that system is reacting. So we can also monitor just water levels. So, and it doesn't, so I'm showing a few analysis here. So you might think just water level gauge, it can really just tell you what's happening with the water level, but you can, there are ways to infer more information from just water level gauging. So we can, what I'm showing here is what we call specific gauge analysis. So essentially that tracks the stage associated with a specific flow over time. So for the same flow rate of 10 CFS at a particular cross section, is the water level now a foot higher than it used to be, or a foot higher than base level conditions? So we can use that to infer bed changes. So without actually surveying the bed itself, we can infer changes, you know, again, using just, you know, readily available USGS gauge data, or even project-specific gauge data. If we have project-specific water level gauges within our system, you know, for that base level period, and it covers a good portion or a good range of flows, you know, we could have the same information, right? Where we can kind of track that sediment pulse through the system. And, you know, this could also be a method. Typically, these kind of specific gauge analyses are more kind of after the fact, you know, tracking something, you know, that has already happened. But there's really nothing that prevents us from using this in a more real-time way. So say, you know, kind of informing the timing of when an initial sediment pulse have moved through the system and the project is ready to release more sediment. I'm kind of referring to a case where you do a dam removal kind of in phases. But yeah, we could use that for lots of different stuff. So water quality. Michelle talked a little bit about this. I'm probably going to not talk a ton about this because we have a presentation specific to water quality and, you know, turbidity and suspended sediment concentration coming up next. But again, these are two water quality parameters that are mostly associated with those sediment releases. So how turbid is the downstream, you know, during dam removals? And again, this comes back to the critical nature of collecting that baseline information. You know, not just at low flows or not just at high flows, but kind of across that range, so we understand the natural variability of some of these parameters, you know, for a better, you know, more realistic comparison to how things are happening during dam removals. All right, I'm going to move ahead. So then, you know, one of the more basic ones, but can be really, really effective at at trying to understand, in a holistic view, how things are changing on site. So taking photos from monumented locations, at specific, either specific milestones or specific locations of interest. You can glean a lot from a photo that you may not have necessarily even thought you were capturing, but you could capture things like vegetation or vegetation changes or things like that. So a plug for really establishing good monumented photo locations. And I'm showing one before and after from the Columbia Lake Dam removal that Beth and some others on the call were involved in. But I know for a fact they didn't just do one before and one after, right? That there's tons of interim photos, kind of showing these progressions throughout. Another plug, you could get the drone imagery again, multiple times throughout these projects, or even move to a video type setup where you're monitoring faster moving processes or time lapse. I think that's it for approaches. I know that was a lot. I'm gonna toss up some references and guidance documents. Again, I think Michelle or Beth, you can correct me. I think we'll probably share these presentations, so there's no reason to kind of scribble all these web links down. But these are some of the references that I've mentioned throughout, and really handy to have around when you're kind of talking through some of this monitoring stuff. I suppose with that, I was going to take questions, but Michelle, you said we'll wait till the end, and I'm fine with that. So I will stop sharing right now and go from there.
Thanks so much, Jake. Yeah, I think we'll save, just for the sake of time, we'll save questions for the end, and we will move right to Krista Reeves with the Musconetcon. Well, there she is, Watershed Association. Yes, and the presentation is being recorded, and we will put it on njdams.org and also on our YouTube channel, but we'll put more information in the chat later on. I'll turn it to you, Krista.
Thanks, Michelle. Get this to present right. Are you seeing the full screen or are you seeing the presenter view?
I'm seeing the presenter.
How do you get it off of that? It's been doing that lately.
In the display settings, perhaps there's up top might be
hide presenter view.
Go to slideshow.
Here, resume slideshow. Hold on. It's got to be some set.
Yeah, if you go back to the PowerPoint and then you're good.
Okay. Now I just need to remember how I did that. All right. Hey, everybody. So I'm Krista Reeves. I'm the Water Quality Program Coordinator at the Musconetcon Watershed Association. And today what we're going to take a look at is the Musconetcon's dam removal project on the Hughesville Dam. Is it advancing? No, it doesn't look like it's advancing.
It is not.
Sometimes if you tip the slide, just press, click on it once, it'll then start to advance.
Yeah. You wouldn't think I'd done this so many times, right? Okay, good Lord. Sorry, everyone.
Sometimes if you go to slideshow up top,
Yeah.
and then or the slideshow tab, rather, not that button, like in the animation slideshow review, like the tabs, go to slideshow. Oop, right next to the animation
Yeah.
field. And then from the beginning, or you could use the turn off the use presenter view. Sometimes that works. Or from the beginning, yeah.
Yeah, it just doesn't want to do anything now. You're not seeing any advance. It's still just that opening slide. I'm sorry, everybody.
No. We're seeing the whole presentation. So I'm seeing all your slides on the left and then the ones that you have highlighted. So it's not in presenter view.
Yeah, all right. All right. Let's see if we can. Third time's a charm, right? I don't know, we might have to deal with this.
It's still in presenter view, but if you
Yeah.
can figure it out, not you, but yeah.
Anybody wants to help out? Anyways,
So if you go to the top, if you can go to the top display settings, click
Yep.
that. And then change your, swap presenter view right there. Yep.
all right. Thank you.
Bill
Good
for the
Lord.
win. Thanks, Bill.
Right? All right. All right, so we're gonna take a look at the Hughesville Dam Removal Project and the monitoring around that. And as Michelle said, there are so many parameters that you can collect to monitor your dam removal that I had to hone in on just a few of them. So we'll be talking about things like the flow, temperature, and fish passage, the monitoring around that, and macroinvertebrate assemblage communities. So this is the view of the Hughesville Dam before it was removed. This came out in 2016. It is a 15-foot dam that went across from Warren to Hunterdon County. And so just we can orient ourselves in space here. So if you can see my mouse, this is 519. This is running across the Musconetcong River. This was the IPPE mill. This is a paper mill that was in production back in the early part of the century. This is where the dam was. And this is coming from upstream. This is going downstream. So one thing I do want to point out that was pretty advantageous for this dam removal is there was these two settling ponds here that was in use for the mill race that was used to help remove the sediment from behind the dam. So this was taken out in 2016. MWA was fortunate enough to have that time to do some pre-monitoring and get some macroinvertebrates and other water quality parameters collected before this came out so that we could compare that to the post monitoring. So this is after it came out. The view upstream is the photo on the left-hand side, and from the actual dam, and then downstream is on the right-hand side. So there was a lot of riprap used, core matting, and lots of live stakes went in. The riparian buffer has not fully grown back in, so there's not a lot of mature trees on either side just yet to help provide canopy. But that's coming in. There's also a lot of phragmites on the one bank that U.S. Fish and Wildlife has been adamantly trying to get rid of and replace, but we all know what a chore phragmites can be. So when I wanted to talk about this dam removal, it brought me to do a flood frequency analysis. So I ran the recurrence intervals from the USGS gauge, which is about 2 miles upstream of this dam. And so I did from the, when the gauge was installed in 1903, all the way through 2018, which is the last year that the USGS gauge had peak discharge values for. I don't know why, there seems to be a lag there, but we'll go with it. So just to point out, what we've got going on here, right? So this is 102 peak flow discharges along this curve here, right? And so to give you a little bit of perspective, this is Hurricane Irene right here at a discharge of about 6,400 cubic feet per second. This one here is in 1979, which was the result of a really bad series of storms in January that also involved a whole lot of snow melt. And also, since Hurricane Ida wasn't available on the latest data, this was also about the same CFS. I have a different visual here for that. So this was Ida. It didn't quite reach that level of 7,200 cubic feet per second, but it was pretty close. And this is Irene right here, just to give a little more perspective. And so what I really wanted to do when I did the flood frequency analysis is I wanted to see how many peak discharges there were prior to the dam removal, and then how many times after the dam removal. Now, granted, there is only, what is it now, 7 years worth of, or 6 years worth of data since the dam removal. But out of those 102 records, there was only 3 that made the peak discharge here. So out of all 102 records of peak, discharge, there was only a couple after the dam was removed. And, if you know anything about the Musconetcong, that part of the river, and before the Finesville Dam came out further downstream, there have been really intense periods of flooding along that lower part of the Musconetcong. And so, also another parameter that I wanted to look at, move forward. Now we're stuck again. Now it's my computer. It's not my day. Here we go. Was water temperature. So we know that, dams slow up the water, they make it stagnant, and that can raise the temperature. So this is from some sondes that Nancy and some colleagues of hers had deployed in the dam impoundment itself. So right, I think 50 feet behind the dam impoundment and in between there, they deployed a couple of HOVO sensors to take, the measurements for temperature over the summer before the dam was removed. And so this is from June through the drawdown on Lake Pacon here in October. So this line right here is the criteria limit for the rolling 7-day average, which is 22 degrees for trout. And so we can see that, there's many times where that is exceeding the criteria limit, and then it drops off here in September. But what is really interesting, and I don't know if she knew she captured this data or meant to, is that you can see when the water level drops, that temperature just shoots right up. At first I thought it must have been, a mistake, or the sensor had come out of the water and that was just riding around in the back of somebody's car for a week until, they got it to the office to offload the data, but that's not the case. So, I went back and looked through the temperature logs and, found the depth measurements here. So the average sensor depth this whole time behind the impoundment was almost 3.9 feet. And so the average summer temperature, taking all this data and just getting a mean average, came to 18.76 degrees Celsius. So if we're looking at it from an average, it was well below what the criteria limit would be. But as we can see, the rolling 7-day average, you have it well above what the temperature criteria would need to be for trout maintenance in summer. And so if we look at that same data today, this is from an Enviro DIY sensor that we have deployed right at the removal site. We see that it is a little bit cooler We still do have those exceedances during the summer on that rolling 7-day average, but the sensor is only 1.75 feet. So the water's cooler, it's much lower. And so once that canopy grows in, we anticipate that, shading it out and then even further dropping that rolling 7-day average temperature in that area. So, it helps to look at all the parameters and the metadata surrounding, yeah, the temperature doesn't look that much better, but let's keep in mind it's half the depth at this point. And so one of the other indicators that we wanted to look at was macroinvertebrate communities. So like I said, the dam came out in 2016, and then we were awarded a U.S. Fish and Wildlife Cooperative Agreement in late 2017. We were able to start our work in the spring of 2018. And so, with the help of these interns who don't look too happy about their picture being taken, we went ahead and we did the work. We went out and collected the macroinvertebrates, and then we sent them off to a certified taxonomic lab to have them ID'd and then the HGMI scores calculated. So as you can see, and this is where the impoundment was, right? So you can't take, very hard to take, macroinvertebrate samples if they exist, other than snails and worms behind a dam. This is right in where the impoundment was, only 2 years after the dam was removed. And we see that all but one had excellent scores. And if anybody was monitoring in the year 2019, they know exactly what happened that year. That was the year that nobody could catch a break from the rain. I think it was the 5th highest year for precipitation in the state since records were being kept. We are only able to take samples at 3 of our 5 locations that we monitor for this dam removal. It was a rough monitoring year, but as we could see, other than this year, either they have remained steady or gotten better. So that is extremely encouraging right at that dam impoundment site. And so the other thing that we, of course, when we talk about dams is fish passage. So we had U.S. Fish and Wildlife, come and give us a comprehensive monitoring plan to gather this fish assemblage data. And then, of course, also presence and absence of some of those anadromous fishes that come up like the American shad. So the method here was the same method where you have 20 points along a certain reach, which is, a downstream netter. And then like this guy with the backpack here, this is basically electricity that goes into the water, and you kind of corral the fish toward the net. You pull them up and you kind of measure and count them. But what we did find that the musconetcong is pretty tricky to do this kind of work. It's pretty much too wide for the seine net to be completely effective. A lot of the touchy fish, which I am told American shad are, will just, as soon as they feel even the hint of that electricity, they run the other way, and you're not going to get a whole bunch of them. We didn't get trout. Some of the quicker fish get away and go around the net. And then again, that high water in 2019 really did impede us from gathering fish data. And then of course, COVID, right? Nobody was able to get into the water. So we have not been able to get all of the fish data that we have wanted to. But from the preliminary data, here's the common catches, and then a pie chart here of how that broke down. And as we can see, we have tons of American eel, which is extremely encouraging since they do migrate up from the ocean. And we have some other, more sensitive species, the black-nosed dace, the tessellated darters that show up over here. So this is really encouraging preliminary data, but we haven't been able to get back in the water since. This is the first year that we're going to be able to get back out there with Fish and Wildlife to do some more electrofishing. And then alongside of that, with our next steps here is some eDNA. Everybody had this time while we were sitting inside, not able to get out there and monitor, and we tried to figure out, well, what are we going to do to be able to get this data? And it seems like a lot of us came to the same conclusion. Well, maybe we can shift focus and, move into the eDNA. And basically what you're going to be doing here, instead of this intensive like netting and electroshocking and needing crews of like 8 people to get out there and catch these fish, you basically are just taking water samples from sections of the river, cross sections. You're going to go across the entire river and filter out the water and pick up the DNA from anything within a certain distance from where you're collecting it, right? So what we're going to be able to do, and I know John Weil is going to be talking about this more, so I won't expand on this too much, is we're going to be able to detect the presence and absence of the Alicine species, which is for us the American shad. And then we're going to be able to get that assemblage data with metabarcoding. And so, we're going to go side by side. Hopefully we can get this all coordinated just right and do the electroshocking with fish and wildlife. And at the exact same time, take these water samples and send them to Rutgers University. And they're going to be able to see, what matches up, what doesn't, maybe something they're missing, help them build that library. And at the same time, fine tune this as a tool that we can use that's way less invasive. You're not going to get as much fish kill, and hopefully it'll be more accurate. Another thing that this is going to be able to do is detect invasive species. So in the Musconetcong, I'll show you a slide in a minute. We have the first reported sighting of the New Zealand mud snail. It is at that dam removal site, so we want to make sure it stays contained, doesn't move any further. In decontamination set up at these access points for anglers to be able to clean off their gear. And so also snakeheads and Chinese pond mussels are of concern in the area as well. And another thing that our next step for us is to install more of these Enviro DIYs. Like Jake was saying, monitor now, right? You got to monitor as much as you can, get that baseline data. So we have set ourselves up. We have one above the Warren Glen Dam, which is the next impediment, fish passage impediment going upstream, and then the Bloomsbury Dam. So we have 2 Enviro DIYs now. What they do is they collect continuous data. Every 5 minutes, they gather data for conductivity, temperature, depth, and turbidity. And with a combination of grab samples and quality assuring the data, we can build rating curves to understand how much sediment is moving through that area. And then so we'll be able to create these long-term data sets with these sensors in the water here to wholly understand what's going on pre and post removal of these dams. So this one on the left-hand side, that's the Bloomsbury Dam. I am told that is going to be starting to be worked on and removed this year. Yay. And then the next one to come out, which is the largest high hazard dam on the Musconetcong, is on the right. That's the Warren Glen Dam. It is a 30-foot dam and or 37-foot high dam. And like I said, it is a high hazard dam, and Army Corps of Engineers has been saying that since 1981. It's always fun to think about your life while you're standing collecting water quality parameters at the base of this dam. You have some existential moments there. And so I would be remiss if I didn't talk about the mud snail. So this is the aquatic invasive that we do have on the lower part of the Musconetcong. I have materials if anybody would like to post this. It is basically just a decontamination poster with QR code that goes to our website to tell you all about this little invasive snail. But with that, I will say thank you. And if you have any questions, we will take them at the end.
Thanks so much, Krista. That was excellent. We do have one more presentation. I know we are almost at time. went a little bit over, but I would like to give John the floor and invite you all to stay past the allotted 2 o'clock hour. We will also, the presenters will also stay on past 2 o'clock to answer any questions. If you do have to hop off though, I invite you to pop your questions into the chat or contact the partnership via the njdams.org with any questions or thoughts if you do have to jump off and you are not able to stay. With that, I will stop and I will turn it over to John Vile with the DEP.
Thanks, Michelle. Can you see my slideshow?
I see your slideshow. It's not in yeah.
Okay.
Okay, good.
Thanks, everybody. My name's John Vile. We're from New Jersey DEP's Freshwater and Biological Monitoring. We typically do fish biomonitoring, fish IBI work. We do some fish contaminant work as well. And we often engage in some special projects, and that's what I'm here today to talk about. Our environmental DNA project we started a couple years ago. So DNA, for those of you who don't know, it's a newer technique to identify trace amounts of DNA present in the environment. It could be in the soil, on leaves, or in our case, in the water. It's a cost-effective way to identify presence of aquatic organisms. It uses a variety of laboratory techniques, qPCR, metabarcoding, to detect trace amounts of DNA in the environment. And this is an important tool for fish biologists to be able to track species of concern, monitor entire fish communities, detect invasive species. So the ways in which fish go about shedding DNA are throughout their slime layer, excrement, scales, gametes, and carcasses also give off DNA. So in 2020, under COVID, we weren't able to work in our normal 4 to 6 person crews to do our IBI work. So we began a little bit of collection of DNA with a consultant in Colorado, Jonah Ventures, and also Rutgers University. And then in last year in 2021, we piloted a project with Rutgers University. We had some goals to develop field sampling protocols. We wanted to work with Rutgers to develop a New Jersey fish DNA library. A lot of times when you send a sample off to have it analyzed for DNA, they'll use these nationwide libraries in which the sequencing is developed in other regions of the US or Canada. And sometimes that gene sequence doesn't always match up exactly. So by taking fin clips from fish while we're out doing our IBI work, sending those to Rutgers, they can develop the sequence for species from New Jersey, provide more accurate results. We also wanted to use metabarcoding to be able to detect entire fish assemblages at our IBI sites. And then we wanted to use qPCR to identify target species. At some of our IBI sites, we have some rare species that are often difficult to collect with electrofishing. And we're hoping that maybe eDNA can be a useful tool for that. So for this pilot project, we decided we're going to use American shad as our target species. These are migratory fish that spend their adult life in the ocean, return to their natal streams to spawn. Since a number of dams have been removed, both on the pollen scale and the Musconetcong, we felt like this would be a good tool to use to be able to detect whether these fish are using these streams. So this is the pollen scale pre-dam removal, the Columbia Dam constructed in 1909, which has blocked the passage of American shad in upstream sections of the pollen scale. And this is post-dam removal. Nature Conservancy and their partners have worked to restore the floodplain and the natural stream channel. And we have had reports of anglers catching adults here and there upstream of the dam. I think there are some electrofishing surveys. And there are some in the state who have, kind of declared that American shad have been restored to the pollen scale. It's a little bit premature. There's a lot we don't know. We don't know if those adults which have been observed are actually staying in the system and spawning, or are they just swimming up, realizing this is not their natal stream, turning around and leaving. This is a common occurrence with migratory salmonids in the Pacific Northwest. The other thing we do know about Delaware stock American shad, based on studies in Pennsylvania and the Schuylkill and the Lehigh, fish that have been tagged, it appears that Delaware River stock American shad do not stray very much. So in other words, if they have a natal stream of the Lehigh River, they typically will return to the Lehigh River and typically won't stray to another stream. So therefore, if the dam is blocked, the pollen scale since 1909, based on what we know from other tributaries, it would seem unlikely that all of a sudden there'd be a large migration of adults into the pollen scale. Now, it is possible there is a remnant population which has been spawning in the lower part of the pollen scale for years below the Columbia Dam, and they're the ones that we're now seeing upstream. We established 3 sampling locations on the pollen scale, one below Columbia Dam, one off of Sipley Road as one of our IBI sites, Vale, and then another one which is downstream of the last dam, Paulina Dam. We have 2 sampling stations on the Muskinecon that we did, one in Regalsville, just upstream of the confluence, and then another station below Warren Glen Dam. So in 2021, we collaborated with Rutgers University to have them analyze our samples. We started with a pole and dip bottle method to begin with in the spring of 2021 for adults. And then later on in the summer, when we were doing some juvenile shad work, we were able to purchase a DNA backpack. We're able to utilize that. So this is a typical pole sampler in which you have a sterilized bottle you attach to a pole. This has a little plunger inside. When you pull a cord, it fills the bottle. This is one of my coworkers taking a sample. The idea is to try to collect that sample as far upstream from you as possible. One thing about DNA, it sticks to everything. It's very easy to contaminate a sample from DNA that's on your wader. So you want to try to do your best to collect that sample as far upstream from you as possible. Obviously, the pole sampling, it's the easiest to collect. Anyone can do it. Take a water sample, send it to the lab. The problem with this type of sampling is sunlight, time, temperature, microbial activity. These all cause DNA in that sample to break down. So you have a very limited amount of time to get that sample to the lab before it needs to be filtered and processed. So again, we purchased an eDNA backpack. This allows us to filter more water, where we're collecting 1 liter bottles per filter with the dip method. We're now, with this backpack, able to collect 3 to 5 liters of water. So we're able to filter a lot more water. This enables us to be mobile, so we can move throughout the stream, sample a number of different habitats, eliminates the need for immediate laboratory processing, reduces any kind of degradation of DNA. These filters are self-desiccating, so once we collect the sample, that filter is dried out, you're able to store it on a shelf or in the freezer for 6 months, maybe a year. Eliminates a lot of contamination. It also presents the option for taking triplicate samples. So at each site, we try to take at least 3 samples. And with a backpack, we can actually collect 3 at one time. So this is my coworker collecting a sample. This is the filter down at the end of the long pole, sucking up through a pump in the back of the backpack. He has a little remote here where he's able to monitor the amount of water being filtered. And once it gives an indication that you've filtered as much and the filter is beginning to clog, You invert the filter, allow all the water to run out, crack this housing a little bit, run the filter for another minute or so to really dry that filter. You toss it back in the sealed bag with the label and it's shelf stable. So just looking at some of the results from our sampling on the pollen scale, this is the downstream location. The way this works is samples 1 and 3 are from the stream margins, and sample 2 is from the sort of the center channel. Unfortunately, the center channel sample was lost. It was actually my fault. I dropped the sample bottle after collecting it. But what we have is significant detection. So each sample they'll run in 3 replicates. So we have detections at all 3 replicates, a significant amount of DNA. So there definitely are American shad in the downstream section. Moving up to this middle section off of Sipley, we have a detection in the main channel and then off to the stream margins. 1 out of 3 replicates, still a significant amount of DNA. It just means that not as many fish, not quite as much DNA in this middle section. And then when we travel to the upstream location, again, we're about a mile downstream of Paulina Dam. We did get 1 detection in the center channel. Again, it's definitely American shad. It's a good hit, but just not as much DNA, likely not as many fish in the upstream portion. Switching over to the Musconetcong, we did get some detections in the downstream locations, but we actually did not detect any shad in the upstream sections. Taking a look at our summer results using the backpack, this is for juvenile fish. Our goal was to try to get out to sample in early July, but high flows prevented us from getting out there. There's sort of a fine line of sampling for juvenile fish where you want to make sure all the adults have left the system, and any adults which have died, their carcasses have been flushed out of the system, because those carcasses will continue to give off DNA. So we felt like early July would probably be a good time period. Unfortunately, we could not get out there because of high flows. We did not detect any shad in the Musconetcong in the upstream portion. We did in the downstream. We had, again, this is off the stream margins where you would kind of expect the juvenile fish to be, often habitats off towards the bank. We have detections in the pollen scale. We did not have any detections in the upstream or midstream, but we did have 1 on the downstream. Very positive results, in my opinion. If you ask me, I definitely believe that these are juvenile fish. I believe that there was spawning going on upstream and that these fish were flushed down with the current. If we take a look at the pollen scale gauge, USGS gauge, this is the time period when we were out sampling. Typically, when we're doing our IBI work, we're running usually less than 100 CFS. And you can see in early July, we're over 600 CFS. So it's pretty likely any juvenile fish that were upstream were probably flushed out of the system in this high flow. And we were just catching the very tail end of the out migration of these fish from these tributaries. So for our 2022 sampling, we want to ramp it up, and we're going to look to do weekly sampling. We're actually starting next Monday. We're going to focus on the pollen scale and do weekly sampling from April all the way through maybe early August to try to catch the timing of adults moving in, spawning, juveniles, and out migration of juvenile fish. For the spring adults, we're going to focus most of our sampling on the main channel, which is where the fish should be moving through. Like I said, we're going to continue sampling into the summer for juvenile shad. And then we're also going to do some spot electrofishing in targeted habitats during the summer. Until we actually physically collect juvenile fish, it's impossible to say whether the DNA we're collecting actually is American shad, young of the year, or if it's a carcass that's in the stream. We're hoping to initiate some mainstream Delaware eDNA sampling this year. with Delaware River Basin Commission. There's some logistics we have to work through with collecting on the main stem. And then we're hoping to continue to support Pennsylvania Fish and Boat Commission with their Young of the Year sampling on the main stem Delaware in August, September, and October. And we want to see if we can try to collect the DNA samples at the same time that we're actually doing that seining, so we can begin to correlate some of the DNA results with actual numbers of fish. And with that, there's my contact information. If you have any questions, go to our website. We have a lot of good fish information, and I can try to answer any questions.
Thanks so much, John. That was great. I invite everybody to, either come off mute, ask your question, or pop it into chat. I do see one in the chat now for you, John. I don't know if you're seeing this as well. Is there a threshold where too much flow render eDNA methods ineffective?
Not that I'm aware of. It's more of if you're in a wadable stream, when can you safely get out there? Our first sampling event last spring we did on the pollen scale. I mean, it was definitely moving. So we don't believe that there's any sort of a high flow. When the flow is higher and you have more sediment flowing down, it's more likely you're going to clog the filters faster, so you may not be able to filter as much water. But as far as I know, there's not a flow restriction.
Great, thanks. There was a question that I received earlier that I popped in the chat, but I don't think it was addressed. And I'm trying to find it to see if other folks, okay, so is the data required for dam removal permitting also required to be submitted to the water quality portal? My answer would be no, but I wanted to check in with others on the call just to make sure that's true.
I don't know if the person asking the question is talking about New Jersey specifically, or if they're talking, for example, about the NRCS portal.
I think they're not on the call anymore. Okay. So hard to say. I was confused as well. If it was for New Jersey, I would say no, right?
Right. I think in New Jersey, if we are approved to submit data, we do submit data. Not that it's necessarily required, unless, as Michelle mentioned earlier, it's required by permit. So for example, turbidity on certain dam removals is a permit condition, the monitoring and the reporting of turbidity during construction. So that would be a case where we are reporting it to the regulator, not via a portal.
Great, thanks, Beth. All right, I see a couple more questions. So one for John. How do you plan on catching juveniles to confirm the eDNA work?
A lot of studies have indicated that the juveniles will tend to congregate in submerged aquatic vegetation. So we're going to focus on vegetation beds and electrofish. We may do some seining, but I think we'll be far more effective electrofishing. And then we also do have some IBI monitoring that we're doing in the pollen scale during that time period as well. So we may actually collect them during our regular fish monitoring.
Great, and I see a question for Jake. Would ground penetrating radar over the pond surface assist with sediment monitoring?
So I am not a GPR expert, but I have seen it used on old cranberry bogs up in the Northeast around Massachusetts. So if you could successfully get it out into some areas, I imagine it would be useful.
I am not seeing any other questions in the chat unless, Beth, did I miss any that you maybe saw or no?
Someone did have a question about monitoring for 319H funding, which I tried to answer. I don't know if that person is still on, whether or not they're gonna have additional questions after my answer, I don't know.
Yeah, if anybody, you could take yourselves off mute, raise your hand, pop it in the chat. We can stay on for another minute or so if others have questions. If not, I would say if you have any ideas, thoughts, interest in additional topics centered around water quality monitoring or really anything, we'd be happy to put together another information session later on this year. We will always take ideas via njdams.org. If you want to, shoot us a email on the contact page, pop something in the chat now. Happy to hear thoughts. Otherwise, I'll put a plug for the next information session. The event has not been created yet, but it will be soon. The information will be posted on our website. The date is going to be April 26th. It's a Tuesday at 1 p.m. And the session is going to focus around the infrastructure bill and the funding that will be coming via the infrastructure bill and how practitioners can anticipate utilizing the funding for their dam removal projects. And if there are no other questions, I just want to take a moment to say thank you so very much to all of our presenters, Krista, Jake, John. That was excellent. As you can see, we went way over because we get really excited about this topic, and there's lots of good information to share and more to come. So keep in touch, and thanks for joining.
Thanks, everyone.